Clinical biomarker studies are often hindered by the availability of tissue specimens with sufficient quality and quantity for molecular assays. While RNA-seq is generally considered the gold standard for measuring mRNA expression levels in tissue, it is a time-consuming process and typically requires multiple FFPE tissue sections in order to extract the necessary amount of quality RNA for subsequent gene expression profiling. The HTG EdgeSeq technology combines proprietary quantitative nuclease protection assay chemistry with an NGS platform to enable the semi-quantitative analysis of hundreds or thousands of genes in a single assay. The assay workflow can be completed in approximately 48 hours and is designed to utilize lysates prepared directly from FFPE tissue. Unlike RNA-seq, the HTG EdgeSeq technology does not require RNA extraction and can use small amounts of tissue material, typically as low as several millimeters, to generate reproducible gene expression profiles. The study presented in this poster was designed to compare the performance of RNA-seq in the HTG Precision Immuno-Oncology Panel, also known as PIP. The PIP assay is designed to measure expression levels of almost 1,400 genes associated with immune interaction and allow for a broad view of the immune response in tumors. Approximately 1,200 samples from across 3 different tumor indications were tested utilizing both technologies. The tumor indications included in the study were colorectal cancer, gastric cancer, and ovarian cancer. Briefly, the samples processed through the RNA-seq workflow were first extracted with the QIAGEN RNA-easy kit using up to 4 FFPE sections, and subsequently processed utilizing the Illumina TruSeq Tumor RNA Access library preparation method. The HTG EdgeSeq workflow utilized extraction-free lysing of a single section of FFPE in HTG lysis buffer and automated quantitative nuclease protection assay on the EdgeSeq platform. Prior to the expression analysis, all samples were evaluated for either pre- or post-sequencing quality metrics to ensure data quality. Only samples that passed QC metrics for both workflows were included in the downstream analysis. Of the samples processed through the RNA-seq workflow, 1,099 generated libraries of sufficient quality and quantity for sequencing. As a result, 91.4% of samples passed the RNA sequencing quality metrics. On the contrary, of the samples processed through the HTG EdgeSeq workflow, 97.3% of samples passed post-sequencing QC metrics, which are set to evaluate RNA quality, quantity, total counts, and assay background levels. For the common genes between these two technologies, the EdgeSeq technology and RNA-seq have similar overall expression distributions. You can see from the image that the majority of data follow a normal distribution around similar values. However, different from the EdgeSeq technology, RNA-seq has a spike at 0, representing values below RNA-seq's limit of detection. HTG EdgeSeq technology and RNA-seq was found to have a good correlation for each of the samples across the common genes. The average Spearman correlation was 0.73. One representative sample from those found to have good correlation between the two platforms illustrates how the correlation of gene expression between the EdgeSeq technology and RNA-seq can be influenced by the spike at 0, representing values below RNA-seq limit of detection. Meaning, although the correlation looks strong across the probes with high and moderate expression levels, the correlation of low-expressing genes can be obstructed by the 0 reads in the RNA-seq. Evaluation of the data sets demonstrated a population-wise comparison between the platforms in addition to a sample-wise correlation. The TSNE analysis, which is a nonlinear dimensionality reduction method of the common genes, revealed similar clustering of the 3 cancer indications between the EdgeSeq technology and RNA-seq, suggesting the methods have comparable ability to differentiate samples from different indications based on the gene expression profiling. One of the more interesting discoveries in this analysis was the appearance of the spike at 0 reads in the RNA-seq data compared to the EdgeSeq technology. To further understand what may be contributing to this, a follow-up study was designed to test the extracted RNA utilized for RNA-seq on the HTG EdgeSeq platform. This study revealed that the proportion of 0 counts in these samples increased from 0.98% to 6.53% on the EdgeSeq platform when compared to the extraction-free lysates of the same samples. This suggests that extraction bias associated with RNA purification may impact the ability to detect low-expressing genes. The HTG EdgeSeq technology detects RNA specimens of 50 base pairs or more, meaning that any short or fragmented RNAs removed during extraction can generate signal and remove bias generated by the RNA extraction. As a result, this technology may offer superior sensitivity.